I'm sharing my screen now. No such. I don't know why that worked on its own. Okay, can somebody confirm you can hear me?
I can hear you.
Thank
I
you.
can hear you.
Thank you. So today what we're going to be doing is just working on chapter 1 again, and then we'll talk a little bit about chapter 10. Any questions about that? All right, we were on this slide last time. Let me remove that. Where we talked about microorganisms being too small to see with the human eye. Come on. I did mention a cell before. Let me go on and state that a cell is the basic structural and functional unit of all organisms. A cell is a highly organized compartment bounded by a thin, flexible membrane that we call the plasma membrane or the cell membrane. All cells contain 3 components. Of course, they have the cell membrane. They also contain DNA and they contain a cytoplasm. You can find all of those 3 parts in all cells. All microbes are cells with the exception of viruses and prions. And we'll talk more about those later in the term, the viruses and prions. If we were to look at these pictures, where do you think you would find a microbe in these pictures? Would you find microbes in the water, on the plants, in the water here, in the air here? Would you find microbes in the plants here, in the ground here, or in the air there? Anyone want to answer that?
You'll find them in all of those places.
Yes, you'd find them in all of those places, on the rock, on the plants, in the water here, in the air. You can find microbes up in the air about a mile into the air. And then in the ground, as well as underneath the ocean surface floor, you can find microbes down about a mile into the soil. Now that may differ if you run into bedrock, solid rock, but with soil, you can go down about a mile and you'd find microbes in it. And then microbes are on this person here, that cat there, on the door or the wall there. You can find microbes in the desert sand, in the desert air also. You'd find microbes everywhere except for one spot in this picture. And that right there is a volcanic vent with magma or lava. And you would not find microbes right here. This temperature would be too hot for any microbe to survive. Any question about any of that? So where you can find them everywhere except for right there. Microbes are almost everywhere on the planet Earth, and we call them ubiquitous. Where is everywhere? Environments where microorganisms are found are on the land and in the water, in the atmosphere. subsurface on the land and under the ocean, on plants and animals. Essentially, wherever liquid water is present, we will find microbes. In fact, you find it when the water is frozen. However, when the temperature is cooler than a minus 20 degrees Celsius, all microbes will be frozen, and they may be alive, but they will not be metabolically active when they are frozen. Any questions about that? So at a minus 20 degrees, we can find microbes, or colder, but they are not metabolically active. And then you can find microbes up to about 120 degrees Celsius. Remember that boiling water is 100 degrees Celsius. So microbes can be found above boiling water, and they can survive that temperature too. I think the highest record is they found a microbe that can be alive, perhaps even growing, at 126 degrees Celsius. And where that is a volcanic vent on the Marianas Trench in the Pacific Ocean. It's the deepest spot in the ocean, and water doesn't boil at that deep pressure with all the water above it until it's 126 degrees Celsius. And they found microbes in that environment. Any question about any of that? All right, microbes are numerically the most abundant life form on the planet Earth. How many microorganisms are there? I'm talking about prokaryote cells. We're talking about viruses and prions. The number of microbes would be about 10 times the number I'm going to give you in this slide. In 1 gram of soil, we have about 1 times 10 to the 8th prokaryotic cells, mostly bacteria, but some archaea and some eukaryotes. Now that will depend on the moisture in the soil and the amount of organic matter in the soil, meaning dead matter in the soil. The more organic matter in the soil, the more moisture you have in the soil, generally the more microbes you'll find. And then the least microbes you'll find would be something like 1 gram of the Sahara Desert soil, very dry soil, very little organic matter in it. Even in that soil, you'd find something like 10 to the 6 microbes or prokaryotic cells in 1 gram of soil. In 1 milliliter of ocean water, which would be 1 gram of ocean water, would you think you would find more cells or less cells than in 1 gram of soil? Anyone want to guess?
More?
More is actually incorrect. The ocean water is one of the deserts of the planet Earth, and you'd actually find fewer microbes in 1 gram of ocean water than in 1 gram of your average soil. You only have about 1 times 10 to the 6 microbes in ocean water. In 1 gram of feces, do you think you'd find more or less than in 1 gram of soil?
More.
Yeah, you'd find more. How about on 1 human being? Obviously, that would be more, and on the entire Earth, you'd have a lot more. So 1 gram of ocean water has about 1 times 10 to the 6 prokaryotic cells in it. 1 gram of soil, on average, about 1 times 10 to the 8th. And like I said, it does depend on the organic matter in the soil and the water in the soil. And the least you'd have would be something like 1 times 10 to the 6 for desert soil and very low organic matter. 1 gram of human feces. If the feces are coming fresh out of the person, you have about 1 times 10 to the 11th cells per gram of feces. Now, if you have fecal matter dried up in the desert, it may be as low as 1 times 10 to the 6 cells per dry feces. And I'm talking about not of a person, but dried up in the desert. Actually, of the feces, 55% of the wet weight of feces is from microbes. On one human, we have about 3.8 times 10 to the 13th cells on us, microbes in and on us. On the entire Earth, the human population is close to 8 billion now, a little less than 8 billion. I could be out of date, but I think it's a little bit less than 8 billion. And on the entire Earth, we have 1 nonillion prokaryotic cells. That is 1 with 30 zeros after it. And I'm sure you've never even heard of a nonillion before, but it's 1 with 30 zeros after it. And large amount of cells on the entire planet Earth. Let me move that out of the way. I can't. Oh, didn't mean to do that. What am I doing here? Somehow I stopped the screen sharing. Trying to get this out of the way. There we go. Now, if that shrinks microorganisms and disease, if a microbe causes a disease, we call it a pathogenic microbe. Most microorganisms do not cause disease. Only a small percentage of microbes are pathogenic, or a small percentage are disease causing. When we're talking about disease, it's important to talk about the difference between an infectious disease and an inherited disease. Can anyone give me an example of an infectious disease? Anyone? What's an example of an infectious disease?
Ebola.
Ebola. Ebola is a disease that can spread from one person to another, and it is an infectious disease. Infectious diseases are caused by microbes, and that's why they can spread from one person to another. That differs from an inherited disease. These are diseases that are caused by the genetic makeup that a person is born with. What's an example of an inherited disease?
Cystic fibrosis.
Cystic fibrosis is an inherited disease. Another good example is oh gosh, I forgot the name of that. Sickle cell anemia. You're born with these conditions. They're not caused by microbes. They're by your genetic makeup. Any question about any of those? In this class, we'll be studying infectious diseases because they're caused by microbes. And they are all infectious diseases because a person can become infected with them. When we're talking about a microorganism, you know they're small. How small exactly are they? They do come in various shapes and sizes. Let me blow this slide up so you can see the numbers a little better. This is something like E. coli, a bacteria that's a little bit bigger than your average bacteria, but not too terribly much. And E. coli is about 1 micrometers long or longer, and a little bit less in width. The average size of your average bacteria is around 1 micrometer. This looks like a fungi, and we can see this is quite a bit bigger. That's 50 micrometers there. And it's hard to make out the cells there, but there's one cell there, and there's one cell there, and this looks like one cell there. Here is what looks like a white blood cell, which is microscopic, but it's a eukaryote cell, and it's much bigger. This one is about 50 to 70 micrometers long. Sorry, my mouse is dying. Because that's 10 microliters there, and that would only go to there. So yeah, about 70 micrometers there. Here's another microbe, and you can see this microbe is quite a bit bigger. That's 50 micrometers there. And this is the microbe. And you'll notice that this microbe, each of the cells of a microbe is one of these dots. And this is a colonial microbe. I used to know the name Volvox, I think. It's the name of this. It grows in colonies, one cell being, like I said, one of these dots. And these right there are microcolonies or baby colonies that are going to be coming out of this parent colony. The parent colony will open up, and then there will be 1, 2, 3, 4, 5, 6 baby colonies come out of it, and then they'll go move elsewhere and establish some place to grow into a bigger colony, the babies. And then this looks like a T4 lymphocyte, and you can see HIV virus being these orange sort of like circles on the T4 virus, T4 cell. That's a helper T cell, by the way. Any question about any of that? All right, so microorganisms play beneficial roles in the planet Earth as well as for people. We already mentioned they decompose organic waste, like in the sewer treatment plant, and when something dies and falls on the forest floor. Microbes are also producers in the ecosystem, and there's two types of producers, the photosynthetic producers and the chemosynthetic producers. Microbes are involved in the nitrogen cycle. We already talked about that. Without microbes, the nitrogen cycle on the planet Earth could not run. Microbes are also used by humans to make various substances, like various industrial chemicals. We use microbes to make ethanol and acetone. We use microbes to make some insecticides. We use microbes to make fermented foods. All fermented foods are made by a microbe. It's the microbe that does the fermentation. And some fermented foods include the making of vinegar. So vinegar is a fermented food. Cheeses are a fermented food. Bread, beer, wine. Another one that you're probably interested in is chocolate. Before you can eat chocolate, it has to be fermented by a microbe. Products used in manufacturing, like cellulase, are made by microbes. Antibiotics, if they're naturally made, they're made by a microbe, although some antibiotics are man-made. And then there's other products used in medicine, like insulin. Nowadays, and gosh, at least for the last 20 years, if not longer, all insulin used in medicine has been made and grown in a microbe. I can tell you that initially, when insulin was first used by a diabetic, they had to purify it from a human. And then they found they could make a lot more insulin if they harvested it from a lamb. So my father, who was a diabetic, initially got human insulin, and then it was switched over to a insulin obtained from lamb. And then later on, gosh, so this is 2003, it's been over 20 years, all microbes came, or excuse me, all insulin came from microbes. So it's probably been at least about 40 years now. Biotechnology is the use of microbes and organisms to produce foods and products for human use. Most people don't realize this, but biotechnology is actually centuries old. We've been using microbes to make sauerkraut and fermented foods for centuries. And so that is using biotechnology to make that product for human. Genetic engineering is a new technique of biotechnology. And genetic engineering first started, I think, in the late 1960s. Became widespread in the 1970s. Applications of genetic engineering, including producing a variety of proteins from bacteria and fungi, like penicillin G, which I don't think that is a protein. I think that's another molecule. Gene therapy, where we can put in a child's defective, repair a child's defective genes by putting in the correct genes into the child. And then genetically modified bacteria, where you put in genes into a bacteria and then use that bacteria to make a product that you would want. Like we can put in genetically modified bacteria to help protect crops from insects or from freezing. Gene therapy, where a child is born with a defective immune system, and then we put in the immune system gene into the child. And then producing a variety of proteins and bacteria and fungi, like when we're making vaccines or enzymes. Microbes have been involved in human welfare, and we call this microbial ecology. Microbial ecology is important because bacteria are important in recycling carbon, nitrogen, nutrients, sulfur, and phosphorus on the planet Earth. And you need to recycle these elements and nutrients, or else they would accumulate in one place on the planet Earth, and then you would never be able to use it for future life. Like I mentioned, that if everything fell on the forest floor and wasn't recycled, then the nitrogen in the plants or whatever fell on the forest floor would just stay there. It wouldn't ever move from one part of the ecosystem to another. That's the same with carbon, sulfur, and phosphorus, meaning that microbes recycle these elements. And it's true that microbes don't move these elements around from one part in the ecosystem to another as much as they do in the nitrogen cycle. But the microbes are important for moving carbon, nitrogen, sulfur, and phosphorus from one part in the ecosystem to another part in the ecosystem. And then they're also important in recycling nutrients. These are examples of microbial-mediated biochemical cycles that support life on Earth. These were first discovered in the 1880s by Winogradsky and Bajorink, showing that microbes could recycle these nutrients or elements in one place in the ecosystem to another, converting like the nitrogen one from one form to another. Any question about that? The reason why Winogradsky and Bajorink were able to discover this is because they, when they were studying microbes, never isolated the microbes into pure species. To notice nitrogen moving around in the ecosystem, you have a microbial community, meaning many different microbes. And one species of microbe can convert nitrogen from one form, like nitrogen gas, into organic forms of nitrogen. Another species, actually several species of microbes, can convert organic matter that dies to ammonia, for example. And they were able to study this because they were using communities of microbes. Most microbiologists did not study microbes that way. They would study a microbe by isolating it as a pure species and then studying the pure species. And the reason why they did that is just because it's much easier to study something if you have a pure species. Then when something happens, you know it happened because of that one species. When Winogradsky and Bajorink were studying like the nitrogen cycle, they weren't sure which of their species was converting, let's say, nitrogen in the atmosphere to nitrogen in organic microbe compound, like pulling the nitrogen out of the air and then putting it into nitrogen in a nitrogen molecule in the microbe. They weren't sure which species was doing that. Any question about any of that? Microbes are important in human welfare, such as in bioremediation. Bioremediation means literally life, and to remedy, meaning to remedy a problem happening for life, like this oil spill. That's an environmental problem, and we can use living organisms to deal with this environmental problem. Like these guys here, they're actually spraying microbes on the oil to digest the oil spill, breaking the oil down into carbon dioxide and water. We use microbes to digest sewage, and obviously bacteria can be used to break down pollutants such as oil and mercury. Now, bacteria can convert mercury from a toxic elemental form to a less toxic non-elemental or organic form. However, it still is toxic. All forms of mercury are toxic, but it would be less toxic if the bacteria can convert mercury to an organic form. Any questions about that? Microbes can be used to make biological insecticides. Microbes that are pathogenic to insects can be alternatives to chemical pesticides. These alternatives are usually not as toxic as the man-made pesticides. And we can use these microbe-made insecticides to prevent insect damage to crops, as well as prevent disease transmission, particularly disease transmission of plant diseases. An example is Bacillus thuringiensis is a microbe that is fatal to many insects. However, Bacillus thuringiensis is harmless to humans and plants and other animals. And there we see this bag of Bacillus thuringiensis, and you can use it as a biological insecticide. And there we see Bacillus thuringiensis right there. My father actually bought this in the 1960s and then used it on his apple trees to prevent the worms from getting on the apples, meaning the worms to get on the apples and then eating the apples. However, he was way too lazy. He only put it on the apple tree one time. And with Bacillus thuringiensis, it needs to be put on the apple tree about every 2 weeks, which is the same amount of time you need to add a a man-made insecticide to treat the apples to make sure they don't become wormy. So it didn't work, but he only used it one time. And another thing is he was growing his apples in Western Oregon. And as you know, Western Oregon and Western Washington get a lot of rain. The rain would wash the Bacillus thuringiensis off the apples. So you should actually apply the Bacillus thuringiensis about every 2 weeks and every time after it rains. And then you would not get wormy apples. My father got wormy apples, but like I said, he only did it one time. And the worms were less that year than if he hadn't treated them at all. All right, any questions about the uses of microbes, either for humans or the use of microbes in recycling nutrients on the planet Earth? If there's no questions, I'm going to switch topics now and talk about the naming and classifying of microorganisms. This is actually mostly chapter 10. We're going to talk about the scientific names. However, this guy right here is actually the first person to come down with naming microbes and other living organisms, coming down with the first scientific names. And he was also the first person to start classifying organisms. Does anybody know who this is? The person who first came up with scientific names and started classifying organisms. Nobody knows who that is?
Carolus Linnaeus.
Carolus Linnaeus, yes. Or Carol Linné is his, I think it's a Swedish name, but he liked Latin so much he changed his name to Carolus Linnaeus to Latinize his name. And I think your textbook calls him, oh geez, let me see if I remember which way. Carl Linnaeus, meaning they Latinized his last name, but not his first name. And I don't know why they did that. Most people either Latinize both names, Carolus Linnaeus, or they go Carl Linné, which is the unlatinized name, his real name. But he changed it to be, I don't know, sort of a scientific name. Each organism has two names by the scientific naming system that Carolus Linnaeus started. And each organism has a genus name and the specific epithet name. An example would be Homo sapiens is a scientific name for humans. Homo being the genus name, sapiens being the specific epithet. It's not really the species name, because for the species, you need both the specific epithet and the genus name. Any question about any of that? This naming system was so good and so widely used by other scientists after him that we still have it today. And we call this the scientific nomenclature or the binomial nomenclature because each species has two names. Bi is two and nomial means name. Any question about that? And just as a side, I'll mention that we still use Carolus Linnaeus's naming system. However, we do not use his classifying system anymore. He classified all organisms into either a plant or an animal. And as you know, there are more things than just plants and animals on the planet Earth. So we don't use his classification system anymore. And the classification system has actually changed over the years. Initially, it was only two things, plants and animals, and then it became plants, animals, and microorganisms. I think they broke it down into four things. Don't ask me what they were. And then they broke it down into 5 kingdoms. And we will use the 6 kingdom approach, which is what they broke it down after the 5 kingdom approach. And there's different kingdom approaches, and we're not going to get into that. Some people use a 9 kingdom system, other people a 15 kingdom system. Some people, I think it's something like a 30 kingdom system. We won't get into the different kingdoms. But let's talk a little bit more about the binomial nomenclature and its system. All scientific names are either italicized or underlined. Like if I were to write a scientific name on the board, I can't write in italics, but I could either type in italics, or if I'm writing on the board, I'd write it out like Homo sapiens and then underline it. The underline meaning that it is a scientific name. The genus name is always capitalized, and the specific epithet name is always lowercase. The scientific names are always Latinized, and they're used worldwide, meaning whatever language you speak, the scientific name for for people or humans would be Homo sapien. So Homo sapien is used as a scientific name in whatever language you speak. The scientific name may be descriptive, or it may honor a scientist. Some examples: Homo sapien. Let me see if I remember this. Homo means the animal or genus walks on 2 feet, and sapien means that it's doubly wise. So Homo sapien literally means wise man. Escherichia coli. Coli is descriptive. You can find E. coli in the colon. So coli is descriptive. Escherichia is actually named after an early microbiologist, Dr. Ascher, and I don't know how you say that name, was an early microbiologist, and Escherichia coli is named after him. He did not discover it. Somebody gave it that name to honor him. Staphylococcus aureus is a name that is descriptive. Aureus is golden, and Staphylococcus aureus is a golden colored cell. It's a sphere, and coccus means a sphere. Staphlo means that these cocci grow in a grape-like cluster. So Staphylococcus means cocci that grow in a grape-like cluster. Any question about any of that? The first time a scientific name is used, it's supposed to be written out, both the genus name and the specific epithet. However, after the first use, you may abbreviate it by putting the first letter of the genus and then put the specific epithet. Like E. coli, it stands for Escherichia coli, and you can abbreviate that E. coli. Staphylococcus aureus can be abbreviated S. aureus. Any question about that? Both of these organisms are found in the human body. Staphylococcus aureus is found, well, it can be found on the skin of about 20% of Americans. E. coli is found in the gut, the intestines, and the colon of all humans. I guess I could clarify all living humans, but it's probably in the gut of most dead humans. Let's see. Oh, I need to say that there is one exception, and that is E. coli is so familiar to everyone that even among scientists, they will occasionally call it E. coli the first time. But in the scientific nomenclature, you should write it out the first time and then abbreviate it. And like I said, E. coli is so common that it is an exception, and that is some people just write E. coli. Any question about any of that? All right, let's talk briefly about the classifying of microorganisms. We can classify all microorganisms into different groups by making a group having something similar, and then the smaller group would be a smaller group that has more similarities. But before we can address this, we need to first take a look at the classification of all living organisms on the planet Earth. So when we have all living organisms, the first classification system would be to break the living organisms down into one of the three domains. So the first classification system is the three domains. We can classify all living organisms into one of three domains: the domain bacteria, the domain archaea, also called the archaebacteria, and then the domain eukarya. These domains are defined by genetic data, meaning we can simply sequence the DNA of a species and then determine which domain the species would fall into. All organisms within domain are more similar in their genetic makeup, meaning the sequence of their DNA is more similar to each member in this domain than any member in the other two domains. So when we're talking about E. coli, its genetic sequence will be more similar to Staphylococcus aureus than it would be to any other species of archaea or eukarya. And that is how the domains are defined, by the genetic data of the organism. Any question about any of that? The three-domain system is also called the tree of life because the species branch off like a three-trunk tree, meaning here's one trunk for the eukarya, and it'll branch off to form the other classification systems. And then that would branch to form smaller classification systems. But what's unusual is this tree has three trunks, and most trees, as you know, only have one trunk. But this tree has three trunks. It is true that the archaea and the eukarya are slightly closer to each other than the archaea or the eukarya are to the bacteria. And that's because the archaea and the eukarya had a common ancestor for a short while that was a little longer than the eukarya had a common ancestor with the bacteria. The difference between these two, and then let's say that one and that one, is very slight. But the point is that the domain eukarya is actually a little closer to the domain archaea and vice versa than either domain is to the bacteria. Any question about any of that? All right, the next classification system, and we're not going to talk about any after this one, is you can break the domain into a kingdom. If you break the domain into the kingdom, we're going to use the 6-kingdom approach, where the domain bacteria is the same thing as the kingdom bacteria. The domain archaea is the same thing as the kingdom archaea. And then the domain eukarya can be broken down into 4 kingdoms: kingdom plantae, the plants; kingdom animalia, the animals; kingdom fungi, the fungus; kingdom protista, the protists. So each of the domains can be broken down into at least 1 kingdom. Eukarya broken down into 4 in the 6-kingdom approach, meaning there's these 4 kingdoms, and then kingdom and domain archaea and kingdom and domain bacteria. Now, you don't have to use the 6-kingdom approach, although the plurality of biologists do use that kingdom approach, but you can use the 3-kingdom approach, in which case each domain is a kingdom. You can also use the 4-kingdom approach, and that is, gosh, how is the 4-kingdom approach? I don't remember. Let's go on to the 5-kingdom approach. You have the 4 kingdoms of eukarya, the plantae, animalia, fungi, protista, and then you have kingdom monera, which includes both the bacteria and the archaea. I don't like that kingdom approach because these are different domains. You should never put a higher order into one lower order. And kingdom monera has 2 higher order classification systems, the domains inside one kingdom. And then there's the 6-kingdom approach. There's also the 9-kingdom approach, which has kingdom bacteria, kingdom archaea, kingdom plantae, kingdom animalia, kingdom fungi, and then it breaks the kingdom protista into a couple of different kingdoms. There's the 15-kingdom approach, which does the same thing. There's the 20-kingdom approach, which starts breaking the bacteria, the archaea, as well as the plants are broken down into 2 kingdoms. And then the protista is broken down into a couple kingdoms. I don't know how many. And then there's higher orders like the 30-kingdom approach. The point is, scientists cannot agree on the total number of kingdoms. Different scientists use a different number of kingdom system, and there is no classification of a kingdom, meaning there is no definition for what a kingdom is. And that's why there's disagreements as to what a kingdom is and which organism belongs in which kingdom. Like I said, we'll use the 6-kingdom approach, which is shown here. Kingdom bacteria, which is also the domain bacteria. Kingdom archaea, which is the domain archaea. And then the domain eukarya split into 4 different kingdoms. But there is no agreement on that. And scientists have actually been arguing for the total number of kingdoms for over 20 years, and they've never come up with an agreement. Any question about any of that? All right. Your textbook breaks the bacteria, the archaea, and the eukarya into different cell types. The bacteria have a prokaryotic cell. The archaea also has a prokaryotic cell, and the eukarya has eukaryotic cells. Now, your textbook gives this the cell type, but some authors call it a superkingdom, meaning the superkingdom would be prokaryotes, And then the other superkingdom would be eukaryotes. The superkingdom under this case is actually all living organisms. Then you break it down into a superkingdom, and then you break the superkingdom down into the domain. We won't use that approach because your author doesn't. I'm just mentioning it in case you see it in another author. And I do use the term superkingdom because that term has been used by biologists long before biologists ever came down with the three-domain system. Some characteristics of the domain bacteria. All of the bacteria are prokaryotic cells. They all have peptidoglycan, a molecule in their cell wall. They usually divide by or multiply by binary fission. They can get their energy needs from any energy source, or an organic chemical like glucose, an inorganic chemical like sulfate, hydrogen sulfide, sulfate, iron. They can also get their energy needs by photosynthesis. Most of the bacteria are free-living decomposers or primary producers, but some of them are parasitic and some of them are pathogenic, meaning some of them can cause human disease. Let's briefly talk about the domain archaea. All of them are prokaryotic cells. They do not have peptidoglycan in their cell walls. Peptidoglycan is a molecule actually only found in the bacteria, the domain bacteria. Some of the archaea are found in extreme environments, and that was the first place we found archaea, an extreme environment. But when we look at all the Earth's ecosystem, we find archaea in every ecosystem. Some of the extreme environments you can find archaea include the methanogens, areas where there's methane, extreme halophiles, where there's extreme salt, like the Dead Sea, extreme thermophiles, where there's extreme heat, you can find archaea. But like I said, when we looked further, we could find archaea in all of the Earth's biomes. Here's a question for you. Are any of the archaea a pathogenic organism? Is there any archaea species that causes human disease? It's a yes or a no.
None of them cause human disease.
That's correct. The archaea, there's no known species that causes human disease, so they are not pathogenic. For this reason, and this reason alone, we will not be discussing the archaea in this class unless I specifically mention them. And after this lesson, we'll hardly ever discuss the archaea because none of them cause human disease. That doesn't mean that they aren't important or that I don't think they're interesting. It's just that the focus of this class is on the microbes that cause human disease, and none of the archaea cause human disease, so we won't study them much in this class. Any question about any of that? All right, some characteristics of the eukarya. Remember, they have 4 different kingdoms: the plants, the animals, the fungi, and the protista. I should mention that the protista is a collection of organisms which are eukaryotes, but they're eukaryote, which is not a plant, not an animal, not a fungi. And so we've always known that the protista is not a monophyletic organization or classification, meaning the protista have different common ancestors. And that's why you can break the protista into different groups, because it's not really a good group. All it is a collection of eukaryotes that is not plants, not animals, and not a fungi. All of the eukaryotes lack peptidoglycan. There's no peptidoglycan in any of their cell walls. And of course, animals don't have cell walls. So peptidoglycan is only found in the domain bacteria. When we're talking about the eukaryotes, we're going to talk a little bit about the fungi. Fungi is plural, singular is fungus. The fungi use organic chemicals for their energy needs, such as glucose. And if you don't know, glucose is the organic chemical that we use for our energy needs. The fungi can be multicellular molds where they grow in a mold mass, or a mushroom, which consists of masses of mycelia. The point is, the mushroom is multicellular, but the fungi can be unicellular, such as the yeast, shown here. All yeasts are unicellular. The kingdom fungi are either decomposers or pathogens, meaning there are some fungi that cause human disease. One that you may have heard of is Candida albicans, which can cause a fungal bloom or a fungal disease in people, like skin fungi, nail fungi, eye fungi. Another kingdom of the eukarya is the kingdom Protista. We can break the kingdom Protista into three different groups: the algae, the protozoa, and then the fungus-like protists. The algae are plant-like protists. They do have cellulose in their cell walls. They engage in photosynthesis to make energy. They can produce oxygen, as well as organic compounds like glucose. So the algae are protists, which are very similar to plants. And some of the algae are microscopic, and then some of them, like the kelp that washes up on the Washington beaches, would be multicellular. The protozoa are the animal-like protists, and they can move, whereas well, actually some of the algae can move too, but there are the protozoa, the amoeba, the paramecium, the euglena. The euglena has a flagella to move by. The paramecium have cilia to move by. And then the amoeba can move, but it moves by, what is that called? Pseudopods, where this cell can pull in the cytoplasm here and then protrude it out there. And then the cytoplasm will grow this way, and then the cell can move this way by moving its cytoplasm, putting it out in a pseudopod here, and then pulling it up here in the back. The protozoa may be motile. They can move by pseudopods, cilia, and flagella. Remember, the domain bacteria, if the cell moves, it's moving because of flagella. because that's the only way a prokaryotic cell can move. But the eukaryotes can move in 3 different ways: pseudopods, cilia, or flagella. If we're talking about a cell, obviously people can move by walking, but that's because we have legs. Some of the protozoa are parasites, and some of them do cause disease in humans. The fungus-like protists we're not going to talk about further. I'll just give you an example of one, and that is the potato blight, a fungus that grows on protist that grows on potato, causing the potato blight. If you don't know, the potato blight caused the Irish famine, which brought a whole bunch of Irish men or Irish people over to America. We're not going to talk much about the kingdom Plantae, but you can separate the kingdom Plantae into the mosses, the ferns, the conifers, and the flowering plants. All members of the kingdom Plantae are multicellular. And the reason why we're not going to talk about the kingdom plants very much is simply because they do not cause human disease. And the focus of this class is to talk about microbes which cause human disease. Let's talk a little bit about the kingdom Animalia. You can break the animals into the sponges, the worms, called helminths, the insects, and the vertebrates. If you do break them down into this, you should realize that this classification of a worm is not monophyletic, and that is the different worms like the roundworm and the flatworm. They are as different from each other in their sequence as they are different sequence of DNA, I should mention, as they are from the sequence of DNA of people, meaning the flatworm is as different from people as it is different from the roundworm. So this system is a little bit human construct, and you really should be breaking this down into more categories, but this is what we use. All the animals are multicellular. Some animals do cause disease, and that are the worms. Some worms are parasitic to humans and cause human disease. Any question about any of that? All right, microbiology and microorganisms. We've talked about microorganism, and we have briefly mentioned what falls under the umbrella of a microorganism. You have bacteria, archaea, certain of the eukarya, like the protozoa, the molds and the yeast are unicellular, so they're microorganisms. Some algae, as long as they're microscopic, they're a microorganism. And then the helminths, the worms, as well as the viruses and the prions, are microbes. But remember, the viruses and prions are not organisms. They do not have cells. They are a microbe that causes disease, human disease, but they are not cells. So they're special. And then the helminths are sort of special too. And that's because we do consider them a microorganism, and they do cause, at least some of them do cause human disease. But most worms aren't microscopic, right? So why are we calling them a microorganism? Like the tapeworm of humans can be, it's either 30 feet or 30 meters, I forget which, can be 30 feet or 30 meters long. That's obviously not a microorganism, right? So why do we call the helminths, the worms, a microorganism. The reason is that the infective stage of the worm is usually, if not always, a microbe. Meaning when you get a tapeworm, how you first get infected is you consume the fertilized egg of the tapeworm, and that's microscopic. Even the schistosomiasis, that disease, the schistosoma worm, actually burrows into the person's skin, and what's burrowing into the skin is microscopic. So the developmental life stage of the worm that is infective to humans is microscopic. Even though most worms are not microscopic, we do consider them a microorganism. And then microbiologists have been studying disease-causing organisms, and some of the worms do definitely cause disease. Now, with that stated, the helminths, the worms, you should realize they are multicellular animals. Many of them are parasitic, but not all of them. In fact, the majority of the worms are not. All of the parasitic worms are either a flatworm or a roundworm. But even of these, not all flatworms and not all roundworms are parasitic, only some of them. And then you should know, I've already mentioned it, the flatworms and the roundworms, even though we treat them together as a worm, a helminth, they are genetically as different from each other as they are different to a human. Any question on any of that? All right, let's briefly talk a little bit about viruses. They are not cellular, so we can say they are acellular. They do have a genome, which consists of either DNA or RNA. But that genome is never both DNA and RNA. It's either DNA or RNA. And that molecule will be the central molecule of the virus. And then that molecule will be surrounded by a protein coat. Some viruses can have the protein coat surrounded by a lipid envelope, but not all viruses have a lipid envelope. All viruses do have either DNA or RNA, and they all have a protein coat. Viruses only replicate when they have infected their host cell, meaning viruses have a host cell they can infect and then replicate in. If the virus gets inside another cell, it's not a host cell, the virus cannot replicate. Remember, viruses are not part of the three-domain system because they are not a cellular organism. Any question about viruses? If not, let's compare the three domains of cellular organisms, the bacteria, the archaea, and the eukaryotes. The cell type for bacteria and archaea is prokaryote. The cell type for eukaryotes is the eukaryote cell type. The chromosome of bacteria is usually one chromosome. It's usually circular, and that chromosome has no histones on it. A histone is a protein that you can find on eukaryote chromosomes, and the DNA actually wraps around that histone protein. So the histone protein is the main protein in a eukaryote chromosome, and the histone protein is lacking from bacteria chromosomes. They do have protein in the chromosome, but they have no histone protein. The eukaryotes usually have many linear chromosomes. And remember, the bacteria usually have one circular chromosome. A circle has the beginning place the same place as the ending place. That's why we call it a circle. The eukaryote chromosomes are linear, like the X chromosome, what I'm trying to draw out here, will have a beginning place and an ending place. And the beginning place and the ending place will be two different places on the chromosome. The archaea is a little bit in between. It usually has one circular chromosome, but on that chromosome, it does have histone proteins. When we look at the ribosomes, we find the bacteria and the archaea have ribosomes of 70S in size. The eukaryotes have a little bit larger ribosome, 80S in size. The bacteria have peptidoglycan in their cell wall. The archaea and the eukaryotes have no peptidoglycan in their cell wall. All right, I think I'll just briefly talk about the history of microbiology. We're going to begin that topic now, and I'm only going to briefly get into it, and then we'll stop a little bit early today. When we're talking about the brief history of microbiology, you should understand that most of that will be over the debate of whether a microbe could spontaneously generate or it came from a pre-existing life form, the theory of biogenesis, meaning the history of microbiology learned concerns this debate whether a microbe could spontaneously generate or whether that microbe had to come from a living organism. History of microbiology includes the golden age of microbiology, the early times of microbiology. Whenever a scientist looked at something, it was usually something new, and they discovered something new. And that golden age happened, you don't need to know the dates, in the 1857 to 1914. I said you do not need to know the dates in this course. You don't need to know any date except for, there's a little bit of history of microbiology. You do need to know this very first date. In the history of microbiology, we can go back 3.5 billion years and find fossil records in the rocks where we can see the first living organism. They were fossils to ancestors of bacteria, and they are the first life on planet Earth. And we have the fossils going back a little bit over 3.5 billion years ago. That's the only date you need to know. Now, the next mention of microbiology was the Babylonians wrote on their clay tablets recipes for making beer. And they did not know that they were using microbes to make the beer, but that is how the beer got made. The next would be about 600 AD. The Mayans used microbes to ferment a beverage from cacao that we would call chocolate today. And you don't need to know any of these dates, but before the Mayans could ferment chocolate, there had to be something earlier, and the Babylonians wrote the first records mentioning microbes, not mentioning microbes, but referring to microbes. And that was because they were using the microbes to make beer. It's just they didn't realize they were using the microbes. And then before that, you have to have the fossil record of the first life on Earth. So you kind of need to know the relative order of things, but you do not need to know any of the dates except for 3.5 billion years ago is the first record of living organism. Any question about any of that? All right, I think I'll stop it here, and we will continue with this lesson next time. If there's no questions, I'm going to end it here, and I will see you in the lab at whatever time the lab has to be. Probably 6:30, I'm not sure. All right. See you in the lab.
Thank you.
All right.